Hello, my name is Dominic Rodriguez. I'm a current junior undergraduate here at the University of Arizona, and I also work in Dr. Lopez's Food and Safety Lab along with my colleagues listed at the top of the poster. The topic I'm going to be presenting is understanding the prevalence and transmission of Cyclospora cayetanensis in produce growing regions in southwest New Mexico. Cyclospora cayetanensis is a protozoan parasite that is linked to the disease cyclosporiasis. This cyclosporiasis affects the intestinal and stomach area of the individual who has it. It is a diarrheal disease worldwide. Symptoms of this disease include diarrhea, weight loss, cramping, bloating, increasing gas, nausea, fatigue, and loss of appetite. Person-to-person transmission of this disease is unlikely because of the sporulation status of the parasite. The only transmission route that we are aware of at the moment is through consuming fresh produce and water that is contaminated with Cyclospora oocysts. Cyclospora oocysts can become sporulated between 1 to 14 days. Fresh produce areas around the world in many countries along with the United States, have recently reported to be contaminated with Cyclospora cayetanensis. As of September 2021, 38 states in the United States have reported cyclosporiasis infections with just over 1,000 individual infections. So the main goal of the study is to develop a better understanding of the prevalence and transmission of Cyclospora cayetanensis in produce growing regions in Southwest, which is Yuma, Arizona, and also Mexico. To conduct this study, my colleagues and I collected 39 samples in the produce growing regions in Southwest and Mexico. The samples were a variety. So we collected irrigation water, surface water, soil, fresh produce, and also floors and handles. The image on the bottom left of sample collection under methods is an image of my colleagues and I collecting a sample from the water canal in Yuma, Arizona. Once we collect our samples from Yuma, and then also our samples from Mexico get sent to us. So once we receive the samples from Mexico and collect the ones from Yuma, Arizona, we then bring them back to the lab and run them through a backflush procedure to get the contents from the filter into a bottle. Most of the time, the bottle is about 500 milliliters. After this, we put the extracted contents into the centrifuge to separate the debris and get a pellet of organic material. After we get the pellet, we put it through a sieve so it can filter everything that is bigger than 125 microns in length. From what we know, Cyclospora is about 8 to 10 microns in length. Once we sieve out the bigger contents, we put the smaller contents through a flow cytometry located in another lab. After this, we put it under a microscope and study the microscopy of Cyclospora and see if it is present. The image that is listed under microscopy is an actual image of Cyclospora and what it should look like under a microscope. After this, we then use a DNA extraction and purification method from the FDA to get the DNA out of the sampled contents. After this, we combine it with 2 primers and probes and also a master mix in order for us to plate the sample on a 96 well plate. We then run it through our qPCR machine to analyze and get the FAM and Cy5 data, which will let us know the prevalence of the 18S rRNA gene based on fluorescence. After qPCR, we put it in a genome sequencing machine to see if we can identify the source of where the Cyclospora is coming from. So far, our samples are still being processed since we have added a couple new procedures into our overall method. For example, the flow cytometry step was developed in order to process all of our samples. The table under results is the type of samples that we collected and also the exact amount of each sample that we collected. So in total, we have 39. One of the other reasons why we collected a variety of sample types is so that we can be informed of potential other transmission routes of Cyclospora to fresh produce. Also, using the flow cytometry machine will help us identify serotypes and GU sample quality for genome sequencing. Another aspect that we are still trying to study also is the life cycle of the protozoan parasite, which this study should help us gain knowledge of. I would like to give some thanks and acknowledgements to Dr. Lopez and my colleagues that work in the lab alongside me, the core facility here at the University of Arizona with the flow cytometry machine, and also WASO. Thank you.